Okay, so now we're going to discuss the prefixes and the suffixes in DNA synthesis. You can skip this, however, I strongly recommend that you know them before we start the lecture. It's going to make things so much easier. So, for you that don't know what a prefix and a suffix is, the prefix is simply something that we put in front of the word and it changes its meaning. The suffix usually comes at the end of the word. Basically, if you know the prefix and the suffix, you'll have a general idea of what the thing is even before we discuss it. So let's start with the first prefix we're going to see, and this is the prefix called DE. Okay, so what does DE mean? DE means lacking, to lack, right? Damn, I have very bad handwriting. So to lack, or it means without, right? So without or lacking what? For instance, decalcification, right? So lacking what? Calcification, calcium. So it's lacking calcium. Dehydration, for instance. Right? Dehydration. So lacking, what's it lacking? It's lacking hydration, which is lacking water. Depigmentation. Depigmentation. Like, you know, some diseases like vitiligo, right? So lacking what? What's it lacking? It's lacking pigmentation. Deamination. Deamination. D, lacking what? It's lacking an amino group. Okay, hydro. So again, and we talked about this, hydro simply means water. Okay, so things like hydrolysis, for instance, hydrolysis. Hydro means water, lysis means, well, to break something, right? So that is it, hydrolysis. Hydrophobic, for instance, hydrophobic, right? Hydro means water, phobic comes from the word phobia, which simply means fear, right? Hydrophilic, Philic means loving, hydro means water, so it loves water. Hydrotherapy, for instance. Hydrotherapy, right? So what's hydro-water therapy? So basically therapy or treatment using water. Number 3, lysis. Lysis, so stuff like hydrolysis, and we talked about this, right? Hydrolysis, so water breaking or breaking using water. Glycolysis, and by the way, this is no longer a prefix, it's a suffix because it comes at the end of the word, right? So glycolysis simply means breaking down the sugar, right? Is, so where do we see the suffix is? And this usually see it in enzymes, so something like polymerase, right? Or kinase, or ligase. Again, this is going to be a suffix. Here's is, right? Basically, always when you see something like this, know it has to do something with an enzyme. And the last one is the O's, right? So basically, when you see this, know that's going to be related to sugars, right? So ribose, for instance. And by the way, again, this is going to be a suffix, ribose. Glucose, right? Deoxyribose, pentose, okay? So O's, you know directly that this is going to be a sugar. So let's recap really quickly. D means to lack or without, depigmentation, dehydration, etc. Hydro means water. Lysis means to break. Is a suffix. Usually it's going to be used something related to an enzyme, right? Now, don't worry about this page. It's simply the core molecular structures that you must know. You don't have to know the chemical structure to it. Just know the formula for it. For instance, when we say hydroxyl, just know that this is hydroxyl, right? When we say methyl, it's called CH3. Amino, for instance, is NH2. Phosphate, we don't even need to know carbonyl, but I don't think we're going to talk about it. Phosphate, PO4, right? Pyrophosphate, you know what? Just pyrophosphate, let's do it like this. This is pyrophosphate. Okay, carboxyl would be something like this, right? COOH, and that's all. Now, you don't have to memorize these, just get a general idea about it. Now, without further ado, let us start our DNA synthesis lecture. Today's subject is going to be about DNA synthesis, and we're going to insert RNA synthesis also with it. Why? Because basically, on a molecular level, they're extremely similar, okay? So, the way we're going to do this is that we are going to take a double-stranded DNA. We're going to start unwinding it. We're going to break it down and keep breaking it down until we get to the core bases that make the DNA. Okay? So, let's start. Now, this is going to be a double-stranded DNA. Okay, here they have, of course, this is a 3' end, a 5' end. Don't worry about it. and the other one also has a 5' end and something called a 3' end, right? So let's take this double-stranded DNA and let's break it down to the core, right? So first of all, let's unwind it. Okay, great, we unwind it, unwind, right? And you have something now like this. Again, it has a 3' end and a 5' end. Let's read it out, make it into, okay. So, first of all, sticking out of it, like this, are things called nitrogen bases. All right, nitrogen bases. Don't worry about it. Don't worry about it. I'm going to talk about this. Okay. Anyway, suppose that we took this. Of course, this is called now a single-stranded DNA. In textbooks, in medical literature, we call it a DNA molecule. Okay, it's called a DNA molecule. Now, suppose that we take, let's say, a strip, of course, nitrogen bases. We've got millions and millions of them. Suppose we take, let's say, 50 of them, right? And so now you have a, well, it's no longer called, I'm not going to draw 50, but you get the idea. Each line here is going to be representing a nitrogen base, okay? And since they're more than 20 to 25, since they're more than 20 to 25, we call this polynucleotide, okay? Now, suppose out of these, let's say there are going to be 50 nitrogen bases here. Let's say, suppose we take, I don't know, like 15 of them, right? So now we have only this small strip. It has 50 nitrogen bases, and now we call it an oligonucleotide. Listen, basically, I've got to tell you what a nucleotide is first. Stay with me, okay? So let's suppose that here there's a nitrogen base. Now, let's suppose we took only one nitrogen base and we put it under a electronic microscope or whatever. This is what we are going to see, right? You're going to see that this is called, by the way, a nucleotide, right? So what is a nucleotide? A nucleotide is simply a nitrogen base that's attached to a sugar molecule that is attached to a phosphate, okay? And we call this whole structure here, we call this a nucleotide, right? Now, suppose that we get rid of the phosphate, right? So we get rid of the phosphate. Now, what's it called? Well, it's no longer a nucleotide. There's no phosphate on it. Now, it's going to be called a nucleoside. Okay? So basically, a nucleoside is simply a sugar that is attached to a nitrogen base. We call it a nucleoside. Okay? So suppose we want to cut it down further and we break something called the beta-N glycosidic bond over here, right? So these two bodies are no longer attached to one another. Now simply this is a nitrogen base and this is a sugar, and remember the phosphate, we cut it off also, and that's that. So the basic building block of the DNA is, well, I can't really say that it's a basic building block, because to make a DNA you'd need a phosphate, a sugar, and a nitrogen base. However, textbooks, they like saying that the nitrogenous bases are the basic building blocks, okay? And so you would imagine that the synthesis of DNA would go something like this. You have a nitrogen base, and it's, it's floating around, it's roaming around, it's looking right and left. Ooh, it finds a sugar, and it grabs onto the sugar. And now we call these two a nucleoside, right? Now, these two are roaming with one another, and then they find a phosphate, and the phosphate goes and it binds to it. Now it's called a nucleotide. So suppose I am the central connector, I am the sugar, right? I first grab a nitrogen base. Now I'm called a nucleoside. Me and the nitrogen base are called nucleosides. And we roam around and we find a phosphate. Now I am called a nucleotide, okay? So how do I build myself? Well, basically, we've got millions and millions and probably billions of nucleotides, right? And they start to attach to one another. So again, this is my phosphate and this is the nitrogen base. I am the sugar and there are billions of me. And then we start to stack on top of one another. We reach the point of oligonucleotide. Once we become more than 20, 25, now we're polynucleotides. And once we form a whole DNA molecule, we get this, a DNA molecule, then it goes and finds a complementary DNA strand, another strand, and you get the double-stranded DNA, okay? Now, of course, this is not enough, so we're going to be talking about the importance of the sugar and some characteristics about it. And I'm not doing this because I like the pentose sugar or the ribose sugar and I'm in love with it and I'm going to tell everyone about it. No. I'm doing this because there's always clinical significance behind it. Then we're going to talk about the nitrogenous bases. We're going to tell you how to identify them, how you, as a medical student, could tell, for instance, a purine from a pyrimidine, or an adenine, guanine, from a thymine, from a cytosine, from a uracil, okay? Okay, so listen, before we dig into the sugar, the nitrogen base, and the phosphate, terminology is important, people. I like terminology. It makes my life much more easier, right? So DNA and RNA. What do they mean? And of course, you either know it or you do like a small Google search and you come back, you say it's deoxyribonucleic acid and this is ribonucleic acid. And of course, you'd be correct. But what does it mean, right? So DNA means deoxyribo. The N stands for nucleic. And the A would stand for acid. Okay, what about RNA? Well, RNA, the R stands for ribo, N for nucleic, and A stands for acid. So, looking at it, you would come to the conclusion that the only difference between a DNA molecule and an RNA molecule is the presence or absence of this word here, deoxy. And if you watch the lecture on the suffix and prefix that we're going to talk about, you already know that deoxy means we're removing something, right? So deoxy is Latin word that means lack or without, right? Without what? Without oxy. What's oxy? It stands for oxygen, O2. Where? On the ribo. What's a ribo? A ribo is a ribose sugar. And again, if you watch the lecture on prefix and the suffix, you'd know that O's is a sugar, right? So RNA, it has a ribose, remember O's, but it is not lacking an oxygen group. And basically, this is the main difference between DNA and RNA, the presence or absence of an oxygen on the sugar, okay? So, and by the way, it's very important when we reach sugar, when we start talking about the sugar, the ribose sugar that makes DNA or RNA, I'm going to tell you exactly why DNA must lack an oxygen and RNA must never lack an oxygen, okay? Don't worry about it. Now, we just want to analyze the word, the acronym, right, and understand what's going on. Okay, so great. So we know where the D came from, and we know where the R came from. They simply refer to the sugar, right? What about N? N refers to nucleic. Why nucleic? Well, in the 1800s, scientists came, they had a microscope, they looked under it, and they found DNA in the nucleus. And so they gave the word nucleic, right? What about the RNA? Listen, the RNA, 30 years later, 40 years later, something like that, they looked under the same cell, well, not the same cell, but you get the idea, right? And they found an RNA molecule inside the nucleus. And so they came to the conclusion that, yeah, this is nucleic, and the DNA is nucleic, right? However, later on, we found out that also the RNA can be present in the cytoplasm. But by the way, if you do not know what a cytoplasm is, this is the cytoplasm, right? This is the nucleus here, the nucleus, and this is just the cytoplasm, Okay, anyway, they found the RNA, it was in the cytoplasm, but you know, they were stuck. Okay, great. So that's N, and that's N. What about A, acid? Well, that's simple. That's because DNA and RNA are acids, right? Not quite. Listen, I'm not gonna dive deep down the rabbit hole on this one, because, you know, I already have like a mini lecture on this. But bear with me, would you? It's going to be fast. Acid versus basic. There are two ways you can think about this. If we're talking about, you know, blood fluid, basically anything physiology, then we know that the pH is going to be less than 7.4. It's going to be acidic, and if it's more than 7.4, it's going to be basic, right? So acidic, basic, that's great, but does not explain the DNA or the RNA why there's an acid here, okay? So there's also a biochemical terminology, a biochemical concept or terminology. Basically, the acid in deoxyribonucleic acid or ribonucleic acid refers to, or it's called acid because of the phosphate group, right? Why? Listen, it's something, it's like pKa, don't worry about it. Basically, the phosphate has the ability, you see it has a positive charge and a negative charge. The phosphate donates its positive charge, and therefore, under, of course, normal physiological pH conditions, it gives away a positive charge, a proton, right? Proton is a positive charge. And therefore, because it donates a positive charge, because it throws away a positive charge, we call it acidic, right? You have stuff like the amino group, right? To clarify this, the phosphate was happy, it was neutral, nothing going on, then it dumped its proton, and now it's called a phosphate PO4 negative, right? Why? Because it donated it, and so this chemical group in biochemistry is called acidic. NH2 is an amino group, same story. It had a positive and negative, it was neutral, it was happy until it found a positive proton, and then it accepted it. And so we call it basic. That's all. So DNA, the acid, RNA, the acid, they both come from the phosphate. Now, I don't know if you noticed this or not, but by simply understanding the name, we now conclude deoxyribo, so there's a sugar, and I really like to change it up a little bit, because I think that it should be called actually deoxyribo nitrogen base acid. Why? Because, you know, this stands for the sugar, the ribose sugar, which is deoxygenated, right? So it doesn't have an oxygen group. And the N for the nitrogen bases, and the acid simply refers to phosphate. That's it. RNA, same thing, right? So sugar, right? The ribose sugar, the nitrogen bases that make the RNA, and the acid, which is simply the phosphate. And that's all. Okay, so this is what the word really means.